But the job's not done, and this is, I think, the penultimate slide. This reminds us that the projects that happen in laboratories are a long way from the projects that end up in the field. In the pharmaceutical, in the development of drugs that would have pharmaceutical application, academics normally pass off their best leads to the private sector to help them develop them for commercial purposes. And that's largely the case in agriculture biotechnology. We know the large companies have been very successful in corn and soybeans and canola and cotton and so forth. But the project that we're talking about here is a project that has little or no commercial value. It's a crop that's important for those who live on $1 to $2 a day. They can buy enough for $1 a day to feed themselves and their families, a family of 4 or 5. But that's all they might eat. And so this has little or no commercial value. So we've done this in partnerships that have started in the U.S. and will, we hope, end in fields in Africa or Asia. But it's a long process. Our basic work actually started in 1988, and it wasn't until 1999 that we had the first transgenic line that looked promising, but it failed under more stringent conditions. Started all over again, re-engineered the plants. Now those have been working for a number of years. We're ready for our first initial confined field trials. These are field trials that are in a confined space that no one has access to, that no material leaves. It gets burned and trashed in that field. If they're successful there, then it goes on to the next stage of trialing in farmers' fields. That can take more time. While you do this, in this reiteration, you must work with the regulatory agencies. Remember that transgenic crops are regulated differently than plants that are developed through classical crossbreeding or through mutation breeding, high-energy neutron bombardment and so forth, and breaking and rearranging chromosomes is something that is approved by the processes, but genetic engineering at this point is not. It's regulated differently. So we as academics have to learn how to do this. This is a very time-consuming process and can be very expensive. And that, in part, is what limits, I think, the involvement of the public sector, such as Danforth Center scientists or scientists perhaps at your institution, from moving into real contributions in agriculture biotechnology. But it takes time, it takes dedication, and specific funding that will allow it to go on.